Hi everyone, welcome back. Today we are going to talk about and processes. This lecture is especially USMLE questions to make it even more interesting and also to be helpful for USMLE candidates. First, we will be talking about DNA unwinding as well as DNA replication. Basically, what is DNA unwinding? So, if this is the DNA, it has like this. This area is known as the replication fork. The question might ask you, or they might either give you this simple and ask you and show you a picture like this. Exactly, you are right. The answer is helicase. After the DNA is unwound, the DNA polymerase 3 adds nucleotides from the 5' to the 3' direction. This is the most important part. There are two DNA polymerases. First, we are going to see about DNA polymerase 3, which will be adding nucleotides from 5' through D3. Now we are going to see how the DNA polymerase 3 adds nucleotides from the 3' to the 5' direction. This is the 5' and this is the 3'. So it adds in the direction from 5' to 3'. Before it starts to add the nucleotides, the RNA primer is necessary, which is shown in the green. It is produced by the enzyme primase. Following that, nucleotides are added in this sequence. This strand, which is produced continuously, is known as the leading strand. On the other side of the replication fork, this is the 5' end and this is the 3' end. Even here, anywhere to start the replication, we need the RNA primer. Following that, we have nucleotides in this pattern. Similarly, but this strand is known as the lagging strand. Similarly, again, when this area of the replication fork unwinds, this becomes a 5' and this becomes a 3' end. And it starts with an RNA primer over here. Starts with an RNA primer over here, followed by addition of nucleotides. Ignore this animation, this doesn't count. First an RNA primer is added, followed by addition of nucleotides. Once this has been done, the DNA polymerase 1, all this while the nucleotides are added by DNA polymerase 3. The DNA polymerase 3 will have addition of nucleotides from 5' to 3' direction, and it will also do a proofreading from 3' to 5' direction. But the DNA polymerase 1 is that enzyme which will be removing the RNA primer. The RNA primer are all removed and the nucleotides are added in that position. Now these strands of DNA are stuck together by the enzyme known as DNA ligase. This is how the replication happens. These small strands which are produced are also known as the Okazaki fragment. The one which is completely formed from the 5' to 3' is the leading strand, and these small strands are known as the Okazaki, and this is the lagging strand. Just a small recap. The leading strand is that which is synthesized continuously from the 5' to 3' direction. Lagging strand is also made from 5' to 3' and which are called as Okazaki fragments, and they are stuck by the DNA ligase. DNA ligase are those which acts upon the Okazaki fragments to form a DNA sequence that aligns complementary with the leading strand. Another thing, the DNA polymerase is what will be synthesizing DNA from 5' to 3' and also do an exonuclease from the 3' to 5'. But DNA polymerase 1 will degrade the RNA primer after the DNA polymerase 3 is done. Then a telomerase is something which will lengthen the end of DNA strands to prevent the degradation. Aging is usually associated with the loss of strand length. But in some cancers, so in order to prevent the DNA degradation, we have telomerase activity is upregulated in some cancers. Coming on to the other topic. What do you understand by topoisomerase 1, topoisomerase 2, and what are the topoisomerase inhibitors? Let's say here. Here this is our DNA, and it is beginning to unwind by the enzyme helicase. Then, but once it starts to unwind, there will be supercoils which is produced in these areas, right? Because this area is getting unwound. In order to prevent the DNA breakage with the help of due to supercoils, we have topoisomerase, which will cut the strands of DNA to relieve these supercoils. There is topoisomerase 1 and topoisomerase 2. So, but if we inhibit these topoisomerase, then the nick or the cut will not be done in the DNA strands. Topoisomerase, we have prokaryotic topoisomerase inhibitor and eukaryotic topoisomerase inhibitor. Prokaryotic topoisomerase is inhibited by the fluoroquinolones so that the organism cannot undergo DNA replication. Similarly, eukaryotic topoisomerase 1 and 2 are inhibited by, one is inhibited by the tecans, irinotecan, and 2 are inhibited by the posides, etoposide, teniposide. They are all the chemotherapeutic agents because we don't want the DNA to replicate uncontrollably in cancer. So therefore, the topoisomerase 1 and 2 inhibitors are used as chemotherapeutic agents in eukaryotes. So they will be, once they ask about the mechanism of mechanism of fluoroquinolones, or what is that drug which will be inhibiting the topoisomerase, then we should know about the fluoroquinolones. But usually in exams, the questions will not be that much straight. The fluoroquinolones are used to treat prostatitis as well as pyelonephritis, ciprofloxacins. And what is the adverse effect of fluoroquinolones? The adverse effect of ciprofloxacin is tendonitis, especially the Achilles tendon. What is a drug interaction that causes? The oral bioavailability is decreased if ciprofloxacin is taken along with divalent cations such as calcium and iron. So the question will be somewhat framed like the drug which is used to treat prostatitis and pyelonephritis and which also has a tendonitis as a side effect. What is the mechanism of that drug? So you need to know that it is a fluoroquinolone and that mechanism of action is topoisomerase inhibitors. Topoisomerase is also known as DNA guidase. Coming on to the ribosomes and the ribosomal RNAs. There are many RNAs. First of that is a ribosomal RNA, which is also known as the rRNA. This ribosomal RNA along with the proteins, they form the framework of the ribosomes. So this ribosomal RNA is a non-coding one, and this ribosomal RNA is produced by the RNA polymerase 1. RNA polymerase 1 produces the ribosomal RNA which will associate with proteins to form these ribosomes. So the processing of the rRNA and into the ribosomes occurs in the nucleoli of the nucleus. That's what we should know. So as I had already told you, RNA polymerase 1 synthesizes the ribosomal RNAs. RNA polymerase 2 will synthesize the mRNA. Let's see how it does. Say for example, say this is the DNA and RNA polymerase 2 acts here and this mRNA is formed. But to be called as an mRNA, it has to undergo certain modifications. Before that, it is known as a heteronuclear RNA. This heteronuclear RNA will undergo certain modifications such as addition of poly-A tail, addition of 5' methylguanosine cap, and splicing of introns and exons. The introns are spliced and the exons are kept together. Once all this is done, this heteronuclear RNA is called as a messenger RNA. But for the messenger RNA to be formed, RNA polymerase is that which is helpful. After all these modifications, it is called as the messenger RNA, right? So all these modifications are happening in the nucleus, and it is exported to the cytosol, where it is translated as a proteins by the ribosomes. But some mRNA can also be associated with P bodies, which are also present in the cytosol. They are nothing but they are docking centers. Say for example, they will be holding up the mRNAs so that it can be processed and stored for the future use. Let's learn about RNA polymerase 2. How does it produce this mRNA? Say this is the promoter region where we will be having the Tata box in it, and this is the mRNA that is produced. RNA polymerase 2 along with some transcription factors will bind to this promoter region, and transcription, which is conversion of DNA to mRNA, will happen few streams downstream, few nucleotides downstream. It will start producing it. So mostly the Tata box is that which is present in the promoter sequence. And RNA polymerase 2 will act on, and along with transcription factors will bind with this Tata box. RNA polymerase 3 is helpful in the synthesis of this tRNA. Now let's come on to another interesting topic about microsatellites and mismatch repair. What is a microsatellite? These are actually microsatellites. They are nothing but they are repetitive DNA sequences which are scattered throughout the genome. So they will be repetitive. They are just not any kind of short DNA sequences. Say for example AGTC, AGTC, AGTC. So they are short sequences which are repeated throughout the genome. What are mismatch repair sequences? These microsatellites, since they are repetitive, there is high chances of causing an error in it. So these microsatellites are usually stable because the mismatch repair system corrects any errors that occur during the DNA replication. So we need to be very clear what are the mismatch repair systems. MSH2, MLH1 and MSH6 are the mismatch repair system. But when there is a error in the mismatch repair system, like anywhere MSH1 or MSH2 or MSH6, then it will develop into cancer, especially the Lynch syndrome. So we need to be very clear about what microsatellites are. They are nothing but short repetitive DNA sequences, usually 1 to 6 base pairs that are repeated. These repeats are scattered throughout the genome, and they are also known as simple sequence repeats or short tandem repeats. They are either called as microsatellite, or they can be called as simple sequence repeat or short tandem repeat. And we also have to be very clear that they are just not some small DNA sequences. They should be repetitive and highly specific DNA